All right now, this is my introduction to the Enzyme Activity Laboratory, in which ultimately you will be designing and carrying out your own experiment on investigating factors that could influence the activity of enzymes. The basis of this assay is going to be to test for the activity of an enzyme called salivary amylase. It's an enzyme that breaks down starch, and it's there in your saliva. We'll be collecting saliva from one of our students, one of the students in your group. Each group is going to have one student donor of saliva. And then in the first week, what we have to do is to really calibrate the experiment, figure out what the correct dilution of the saliva is that will be giving us an optimal opportunity to detect the kind of results we need. And what I'm doing with this video is to kind of introduce the layout of our experiment and the concept of dilutions. Once we have the experiment calibrated and ready to go, then we'll be deciding how we're going to be setting up our experiment for next week. All right, so here we go. This is a microtiter array. You'll notice that it's a plastic, non-disposable tray that has 96 wells. There are 12 rows with 12 columns, or I think we've got 8 rows of 12 columns, and each row contains 12 little wells. And so if I turn this sideways and do a little diagram, you can see that there are 12 individual wells there. And so this experiment is going to start off with our putting 1 drop of Lugol's iodine in each of the wells in this row of 12. And remember, Lugol's iodine is something that will detect the presence of starch if it's there. We haven't added starch yet, so it's kind of brownish, and if we do add starch, it should turn kind of blue-black. All right, so there's going to be more instructions on how you collect your saliva. There are safety rules. Refer to appendix. I don't know which appendix it is, but there's an appendix in your lab book that talks about the safety procedures for handling body fluids, and we'll be going through instructions later on how we'll be diluting the saliva and collecting some type of diluted enzyme. Okay. So what we're going to be doing here is we'll be starting off with a mixture, and so that's not the one I wanted to show you. This is the one I wanted to show you. Okay, and so it's going to start off with a test tube that's got 25% starch, 2 mLs of that, some pH 7 buffers, 2 mLs of that. The starch is basically the raw material that will be starting off with a substantial amount of starch, and ultimately the enzyme is going to be breaking that starch down to the point where it's no longer detectable by Lugol's iodine. The pH 7 buffer is really there to maintain a consistent pH in our enzyme system. Basically, enzymes, if they have a preferred pH, this will give us an opportunity to investigate the effects of pH on our enzyme later on. pH 7 is a nice neutral one. It's more or less what we found the pH of saliva to be. And then we'll have 1 ml. This is actually a little bit different from what it says in the handout that you guys got earlier. In the handout it says 2 ml of enzyme dilution. I'm making an executive decision here to say, well, it's going to be 1 ml. And so your diluted saliva is going to be your enzyme dilution, and you'll be putting 1 ml of that into this tube. You'll be putting a cap on top of that, some type of parafilm. You could put your thumb on top of the parafilm and invert it a couple of times, and what will happen is that you'll end up with a nice homogeneous mixture. It shouldn't take more than a couple of seconds to get that. And immediately upon getting that, immediately upon getting to the point where you've got the enzyme completely homogenized, we're going to be putting 1 drop of that into the first tube. So this is going to be like 0 seconds. At 0 seconds, we're going to be seeing obviously a color change. Remember, there's going to be starch there, and the effect of starch and Lugol's iodine would be a change to blue-black. Okay, so we'll see if I can draw that. Okay, so there, yeah. If you add at 0 seconds, it should turn blue-black, and then we'll be doing this again every 30 seconds. So somebody's going to be there watching the clock, and at 30 seconds we'll be adding a drop from that same tube. The same tube is going to be taken. Now the enzymes have had 30 seconds to work on the enzyme, on the starch, and maybe the starches are still there, maybe they're not. And so after 30 seconds, we might get uh, another if a positive result. Basically, by getting a positive result, we know the starches are still there in the solution. And so we keep on doing this. We'll go every 30 seconds, 60 seconds, 90 seconds, etc., and add the the starch, the enzyme starch buffer mixture, and we might be getting results. And so this would be a positive result, positive result, a positive result. And at a certain point, though, we're going to be having having the starches completely broken down by the enzymes. And when we get a result that doesn't give us any change at all, in other words, you add the starch, or what used to be starch and enzyme and buffer solution into the Lugol's iodine, and we get no color change, this indicates that the enzyme has completed its activity. We've done away with all the starch in the solution. Okay? And you might want to run that for an extra 30 seconds and see whether we get the same result again. And if that happens, we could just pretty much confirm that we have no starch. That would be, let's see, we would have starch at 120, but by the time we get out to 150 seconds, there's no more starch in the solution. 180 seconds, and so at that point we don't need to to finish the rest of the assay. And so this is going to be the basic layout of our experiment. We would determine from this that it takes about 150 seconds for the reaction to complete. Okay, this will be the tube that is, I think it's labeled ET, and you'll be running another tube that is effectively the same, except instead of 1 ml of enzyme, you'll be adding 1 ml of water and be running the same experiment. And obviously in that case, the expectations would be different, right? If you add only water to the starch, and so if you add only water to the starch solution, in other words, instead of this enzyme dilution, if you had water up here, there wouldn't be anything to break down the starch, and you would probably expect to get positive results all the way out to the very end. And that would be your negative control, or the NC tube as it's described. Okay, so that's the basic layout of our experiment, and this is the way you'll be testing for the activity of the enzyme salivary amylase in the enzyme dilution that you've collected. Okay, so now that you understand that, I guess a couple of things. First of all, obviously, the stronger the enzyme, the less time it's going to take for the starch to be completely broken down. Okay, and the weaker if the enzyme activity is lower, it would obviously take a greater number of seconds, and that's how we're going to be detecting the enzyme activity, or that's how we'll be quantifying the enzyme activity in this experiment. All right, now having said that, the enzyme dilution, this thing that we have here, is it's kind of like the important point. This is our big question mark. What is the appropriate strength of enzyme that we need to start off with in order to make this experiment go? What we would like to do is we would like to see somewhere between tube 4 and tube 7, somewhere in this range. This is where we want the color change to take place. This is going to be like our default setting. You know, somewhere like 90, 120, 150, 180. Anywhere in that range, that would be a great place to start and have our baseline for enzyme activity. So if we do something that makes the enzyme a little bit more active, we would see a decline in the amount of time that would take for starch to break down. If we do something to the enzyme that made it less active, we would see it require a greater amount of time. Okay. Now I'm here to tell you that it's not that easy to find this correct dilution, and it varies tremendously from student to student. And so having run this a couple of semesters, I can tell you that ultimately we find that the correct dilution is somewhere between 1 in 50 to maybe 1 in 150, but it can be as low as 1 in 20, and it can be substantially higher than 150 as well. I've seen that as well. So there's not one solution, there's not one protocol that I can give to everybody and make this experiment work every time. Depending on who the donor is and who the donor is, it's pretty much the main variable. We might need to dilute more severely, or we might not need to dilute very much at all. Okay, salivary amylase is one of those enzymes that's not especially important. If you had no salivary amylase at all in your spit, it would probably have very little impact on your ability to survive and reproduce. Okay, so let me zoom back out to the appropriate size. I think it was at 76. And yeah, so let's go back out to here and think about how we're going to be doing these dilutions. So first of all, let's work with the dilutions that are there in your textbook or in the handout. So let's say we have a solution of so this is your saliva. Okay, so saliva, you collected 8 ml of saliva. This is done the right way. There's no bubbles. You've gotten rid of all the excess debris by rinsing your mouth out really well, and so this is pretty much pure bubble-free saliva. Now, according to the protocol, this would be full strength. Full strength. Now, according to the protocol, you would take 1 ml of this, and you would add that to 2 ml of pure water. Okay, 1 ml of full strength saliva plus 2 ml of H2O would give us 3 mLs total, of which 1 ml is actually saliva. Okay, total. And so to calculate the dilution factor, you basically divide this number here, the full strength saliva, by the total amount of the total volume there. So this would be a 1 in 3 dilution. The dilution factor is 1/3 or 3, depending on how you break things down. So 1 in 3 dilution would be what we're starting off with. Now, if you go back to this picture up here, you'll notice that we're taking 1 ml of 1 in 3, L of 1 in 3 dilution, and adding that to 4 mLs of other stuff. Right, we've got 4 ml of the pH 7 buffer, actually 2 ml of that and 2 ml of the start solution to give us a total of 4 ml. We had 1 ml of our enzyme dilution to that. So what we're looking at here is a total of 5 mLs, of which 1 ml is a 1/3 dilution of full strength. Okay, so if you do the math here, 1/3 dilution relative to full strength, diluted again 5 times, this would give us an overall dilution factor of 1 in 15. See how I got that? We started off our dilution factor and said, well, here is our full strength. We're going to dilute that 1 part in 3, and we take the 1 part in 3 dilution and make that and dilute that 1 part in 5. So basically, we have a 1 part in 3 times 1 part in 5 dilution factor. This gives us a total dilution factor of 1 part in 15. So inside of this tube here, we basically have 1 part of saliva for every 15 parts of total dilution. Okay. Now I could pretty much guarantee you that 1 in 15 is going to be too strong. In other words, with 1 in 15, you'll probably have a color change, maybe in the first tube, maybe in the second tube. But the strength of your saliva, the strength of the enzyme in your saliva is probably going to be too strong with a 1 in 15 dilution. If you wanted to exercise some foresight, you might say, well, somewhere in the ballpark of 1 part in 70 to 1 part in 120. That's usually, this is usually the ballpark that we have to hit in order to get the correct range, basically hitting it from 90 seconds to 180 seconds, right? Okay, so yeah, if we were to dilute further, we could take our 1/3 dilution factor. Remember, we're going to be diluting it again 1 part in 5 later on. So if we for example, we wanted to shoot for a for a stronger dilution, what we could do if we wanted to shoot for, let's say, um, a 1 in 50 dilution. Okay, if this is our target. What you can do is you can start off with a 1 part in 10 dilution, and knowing that we're going to be multiplying it by 1 part in 5 when we add it to the starch and the buffer, 1 part in 5. This would give us the 1 part in 50 dilution that we might be shooting for. So in other words, instead of starting off with a 1 in 3 dilution and diluting that 1 part in 5 to get 1 in 15, we could start off with a 1 in 10 dilution, dilute that 1 part in 5 to give us a total dilution factor of 50. Okay? And how would you do that? Well, it would be pretty easy. If we got a 1 in 3 dilution by taking 1 ml of full strength and adding it to 2 mLs of water to get 3 mLs total, we would get a 1 in 10 dilution by taking 1 ml of full strength plus, sorry, that should be a plus, 9 ml of water. And this would give us 10 ml total. And 1 in that 10 ml would be full strength saliva. So you could do the dilution factor. You could do the dilutions that way. 1 ml of full strength saliva, 9 ml of H2O, giving us an overall dilution factor of that of 1 in 10, and that times 1 in 5 dilution, the overall dilution factor for that experiment would be 1 in 50. So what you can do here is, if you set it up the way that we've described it, you can say, well, with a 1 in 15 dilution factor, we got a result that where everything changed within 30 seconds. That was too fast. We could do another assay and say, well, at a 1 in 50 result, we got a result that we got something that took 90 seconds, which is exactly right, right? And if you get that result, you could say, well, now I know that next week when I do this experiment, I will want to start off with a 1 in 10 dilution factor because I'll be diluting that 1 part in 5 to give us an overall dilution of 1 in 50. And this might be for, this is very specific for the individual whose saliva is being used. So if Roberta is the saliva donor, then you'd say, well, for Roberta, you'd need to have a 1 part in 50 dilution in order for this dilution to work. Okay, so that's the way this works. Now, there's another thing that you might be aware of, which is that you don't have to always go back to the full strength saliva to set up the original dilution. For example, okay, let's say you do a 1 in 10 dilution factor, which would actually give us an overall dilution factor of 1 in 50. And you found that this was still too strong, right? And so you want to shoot for a 1 in 100. And so if you do things the way that we've described it so far, you would probably have to go back to the full strength saliva and set up a 1 in 20 dilution in order to make that work. You don't have to do that because you can get a 1 in 20 dilution by taking the 1 in 10 dilution, and maybe you could take 2 ml of that, and you could take plus 2 ml of water, and what you'll have there is this would be a 1 part in 2 dilution. Basically, this is going to be a 2 parts 2 ml of this 1 in 10 over 4 ml total of solution. Okay, so this would basically be if we do this, 1 ml or 2 mLs of the 1 in 10 plus 2 ml of water, this would give us a 1 in 2 dilution of the 1 in 10, and that would give us a total dilution factor of 1 in 20. And that, if you were to follow the protocol that we have up here, take and do the 1 in 5 dilution at this stage, you would end up with an overall dilution factor of 1 in 100, which would push the reaction time over to the right by some amount. Okay, and so yeah, if you find that 1 in 100 total dilution is too strong still, you might want to push it to 1 in 150, and you could get that by taking setting up a 1 in 30 dilution and saying, well, that's going to be diluted again 1 part in 5. And that will give you an overall dilution factor of 1 in 150. What you want to do with this week's experiment, with this week's exercise, is to find out what exactly is the dilution factor that will work best to give you this baseline result of a color change intermicro titer assay somewhere between 90 seconds and 180 seconds. Okay. I know it's probably going to sound real mathematical and confusing to some of you, but you might want to listen to this again. If that is still confusing to you, then I'll try to straighten things out before we actually start this essay on our laboratory this week. All right. Um, oh, and once we get this, I mean, what I'm hoping is we'll be able to finish this well before our 3 hours is up. And once we're done and we have this result, we pretty much know exactly the strength of the dilution factor that we need for the following week's assay. Then you'll be making your plans. Some of you are going to be working on the effects of pH. You could be thinking about, well, how does pH affect the overall activity of your enzyme? Some of you will be working on the effect of salinity. You can adjust the total salt concentration in the enzyme system. Some of you will be working on temperature, so you can adjust the temperature in which the enzyme is having its activity on the starch. And that should also have some kind of impact on the rate of the enzyme catalysis. And so we'll get to that part at the end of our lab meeting. What we want to do is kind of get the ball rolling initially, and at this point, I'm hoping that you've got at least a little bit of an idea of how we're going to be working out, how we're going to be achieving this goal of knowing exactly what to do in our following week's experimentation.